So in that live, no one is there, but still, I in that live I will explain about the revision of the plant physiology. So what you will study or revise about that in the plant physiology, like those are in the biotech student or microbiology student, it's been very difficult to cover all the syllabus of that plant physiology, but still those are the those are preparing for that exam, so they should study about that because a lot of questions are coming from that part. So like in the physiology, what is the plant physiology, and you heard about that the branch of biology that deals in the how the living organisms grow, develop, and their functions. So they are interact with their physical and their living environments. So that is the kind of physiology, and when you study about the plants, and there is a plant physiology. They are major components. On what are the major components? The major components are the like photosynthesis in the chloroplast, gas exchange, and in the leaf, and also the water conductions in xylems, and the ion transport across the membranes, the signal transductions and pathways, and involving their light harvesting, and also the some of the like hormones and how they work and their genetic expressions during this development. So these are a kind of key important factor for the plant physiology part. So like in the introductory part, and what are the more significant part, you can say like the photosynthesis, like their physiochemical process. This is a kind of, because in photosynthesis you get like synthesis of the organic compounds like carbohydrate, and with the help of the light energy, but kind of more specifically light energy is more responsible for the synthesis of the carbohydrates. So that's why it's important. So today this live session only those are the important topics, like so what are the significance of photosynthesis. You can check out that. That's very easy things, because they are need the food. The only source of the food is the primary source on the food on the earth, that kind of significance. Or you know that the oxygen is needed for the animal kingdom. So like the also atmospheric photosynthesis, that this kind of thing. And along with that, the some early discoveries are there, not so important, but still for somehow some descriptive questions it's important. You can go through that kind of things, like I'm just giving some of these example, like Joseph Priestley discovered the plants producing oxygen in 1773 at that time. And then Julius von Sachs and the T.W. Engelmann, there's a kind of fast photosynthetic action spectrum was discovered by him. That's one of the key important. And also so three name you can just remind of that. And the experiment of this Engelmann, you can check it. That's not too difficult experiment, but you can go through that. And along with their Cornelius Van Niel, that's the discover the fact the photosynthesis is a light dependent process, and also the compound get oxidized and giving the hydrogen out and reduce the CO2 to the carbohydrates. So these are important for this Cornelius Van Niel. Or like the observation is that like CO2 and the hydrogen sulfide, yes, CO2 and two molecule of hydrogen sulfide, and that can make the CH2O and the water and the sulfur. So like their oxygening bacteria mainly accumulates near those segment of the algae, and they are receiving and the red blue components of this electromagnetic spectrum. That's the thing. So you can go through that. And also one curve is there. In that curve you can check out that the absorption spectrum and the action spectrum of that, because their wavelength and how the absorption spectrum and their action spectrum, where it is a kind of T.W. Engelmann experiment, not too much important, but you can just go through that. That's not so much important. So let's talk about their next experiment, like Ruben Kamen and the Hasid experiment. In Ruben Kamen and Hasid experiment, you just remind that like carbon dioxide, and they take the heavy water like H2O16, O16 or O18. That's a, this is kind of taking the oxygen's isotope. Okay, so and with the presence of light and chlorophyll, it can produce the glucose, water, and the isotopic oxygen because they take their heavy water. So, like photosynthesis in the presence of the isotopic carbon dioxide. So, and then this carbon dioxide into water, it's formed their glucose and the heavy water and oxygen. So, in 1940, this experiment, just remind the date, the 1940 Ruben and Kamen experiment. They provide that the O2, it's evolved during the photosynthesis and that's come from the H2. This statement is very important. This is the statement, like this comes from the O2 is coming from the water. That is the simple statement coming from the 1940 experiment of Ruben, Randall, Kamen, Height. So just 4 names are there. You can get puzzled, but still that is the important. And then another important aspect is the Robert Hinn and the Bendal experiment. Like the proposed J scheme, I think we'll go through these complete details not in another diagrammatic video. And in that revision part, you just go through that and how this kind of organisms involved in the photosynthesis. So like you already know about that the 6 molecule of carbon dioxide and that can bind with the 12 molecule of water, and their light energy comes and then they make their glucose molecules along with the 6 molecule of O2 and the 6 molecule of water. So that is the organisms, and like photosynthesis is performed with the presence in the presence of the light energy and like okay. So then we go through the like they are anoxygenic photosynthetic organisms, like in somewhere like in that the involves of the prokaryotes or the green purple photosynthetic bacteria. They do not release their molecular oxygen as a byproduct because they don't utilize their water in their source of electrons. So let's go. I'll not go through the details. Today is a kind of revision what you have studied already, just making the storyline of your study, and that can help you to remind all the stories label one by one. So let's talk about the photosynthesis. Now you can check out the oxygenic photosynthesis and the anoxygenic photosynthesis. And in oxygenic photosynthesis is like kind of prokaryotes to cyanobacteria or eukaryotes to the green plants or photosynthetic protist. And when you talk about the anoxygenic photosynthesis, that's kind of prokaryotes to green and purple photosynthetic bacteria. So that's you can separate these 3, like cyanobacteria, the green plants of photosynthetic protist, and there another protein like green and purple photosynthetic bacteria. Okay, so hope I give that the revision portion, and then you have to remind 4 important things like photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. Like 4 things. Photoautotrophs, where the light is needed and carbon source is CO2. Photoheterotrophs, their light is also important, but carbon source is a organic compound, not CO2. When you talk about that chemoautotrophs, the energy will be the chemical molecule, but source will be the carbon dioxide. And when you talk about the chemoheterotroph, chemo, it means the energy source is the chemical molecule, and heterotrophs, it means their carbon source is from the organic compound. Hope that You can memorize these things, or like, because in somewhere if you want to separate with them, that can be important. Now you talk about the site of photosynthesis. You all know about that, where it can form, and then one of the key aspect of chloroplast. And hope you read the history about the, like, more of this proposal theory of chloroplast or mitochondria related things, but not for the csir, because it's not necessary. You have to read about that. So now we talk about the chloroplast and what are the important aspects of the chloroplast, like the functions of stroma. Their dark reaction is there, and why is the dark reactions, like CO2 into sugar and NADPH form in the light reaction. Sorry. Okay, let's talk about one by one. It's a dark reaction, and in the light reaction, like, they are trap the energy light in their molecule. That's the that thing, light reaction, like synthesis of ATP and NADPH. These are called the light reaction. And when you talk about the CO2 into sugar and with the help of ATP and NADPH, matlab CO2 to sugar, that is a dark reaction, because there is no such kind of involvement of light. That's why dark. If the light is harvested and changes to the ATP and the NADPH, that's why it's a light. So where will be light? Light will be in thylakoids. You have to read about the structures about that. Hope that you read it. I'm just recalling your memory to that. Like in stromal thylakoids to light reaction, ATP and NADPH production, and in that stroma region, it's responsible for the convert of their CO2 to into sugar. That's a dark reaction. Hope that you guys remind all those things. So in that recall part, what are the key important aspects? ATP, NADPH, CO2 to sugar. That means use of the ATP. That's the thing. Now you think about the what are the another key important things, like chlorophyll, bacteriochlorophyll 2, and carotenoids and their phycobilins. So these are the kind of pigments found in the plant body. So you are studying about the physiology of that. That's why you have to more important and give the more significance to the chlorophyll, bacteriochlorophyll 2, because you have studied and making some comparison about that, and the carotenoids. It's the important aspects of plant, and the phycobilins. Let's talk about the chlorophyll 2. So one important key aspect you have to remind about that is the porphyrin ring. So like this is the kind of, these are kind of lipid molecules, are soluble in a organic compound but insoluble in the water. So lipid molecules are soluble, but the organic compounds are insoluble in the water. So it present all the oxygenic photosynthetic organism. They are called their tetrapyrrole compound. That means they are kind of porphyrin derivatives. Tetrapyrrole compound. So you have to remind about the tetrapyrrole. Okay, because this physiology part, you have to remind all those things, kind of memorize all those things. Not too much concepts are there, but still it's a physiology. That means some of the concepts are there. But for exam purpose, I think for my perspective, you can say you have to recall these things. Then your understanding will be better and you can give the answers confidently. So like there are many type of chlorophylls are there, kind of chlorophyll A, chlorophyll B, chlorophyll C, and chlorophyll D, along with the chlorophyll F. So A, B, D are chlorine derivative, while C are porphyrin derivative. So like one of the important thing, like magnesium ion is present and the center of the tetrapyrrole ring. If I want to draw it, I'll practice. You will practice that. That the thing there. How to draw the porphyrin ring, because in example it's not needed, but still you have to just practice about that. and some of the central atom is the magnesium that you have to recall. Magnesium cannot be utilized in any other reaction taking place in the cell. So this is the important aspect. So magnesium is needed in the chlorophyll section. So in pyrrole ring 3, it's attached in a cyclopentane ring, ring V. So if I draw that structure, that time you can understand. Otherwise, it's very difficult to understand. But still, you have to just go for that. So like this is the our ring structure. Magnesium is there, bonds are there. So you have to remind two things. One is that is a cyclopentane ring. So you have to think about the cyclopentane ring of 4. Whether one propanoic acid is attached and with the ester bond to the phytol. Then it will be like the structure of the chlorophyll molecule and showing ring 1 you can show. If I give presentation in another thing, then you can recall it like phytol attached in cyclopentane ring of 4. That you have to remind. Then it's about chlorophyll fine. Then another chlorophyll A, chlorophyll B, chlorophyll C. You have just to go through it. It's not too important, but that's the important thing. But just to remind it. And carotenoids, that's very important. Kind of thing that except the eubacteria, carotenoids are there, and these are kind of linear terpenes, tetraterpenes. Yes, they are linear tetraterpenes, are the hydrocarbon and conjugated with the double bond. Okay, fine. And then they are these are the carotenoids and carotenes like C40H56. Just remind it. C40H56, they are kind of empirical structure of the hydrocarbon backbone. Alpha carotene, beta carotene and gamma carotene. 3 types of carotenes are there, responsible for the orange color. And most common carotene are, you know about the beta carotene.